Hi, this is my active recall practice to make it stick for week 5 of Psych 3310. And this is the first part, so the vertical pathway for the retina. So I'm going to be taking from top to bottom, like a top to bottom approach, where I look at the eye first and then we go all the way to the graded potential. So first we start with the eye, and then we go down to the anterior surface, which is called the fundus. And what we see in the fundus is the retina, the optic disc, the fovea, and the macula. And this is how we get the vertical pathway. And the vertical pathway is just where like a light photon hits the photoreceptor, and then from the photoreceptor it goes to the bipolar cells and then to the ganglion cells, and then goes through the brain via the optic nerve. And basically what we need to know is how it just goes through those like steps, step by step. So first we need to understand what the photoreceptors are, and that's the first step. That's what like gets the light to converge onto other things. And that's basically when we get the photon to hit these photoreceptors, that's what makes us send the signal past onto the bipolar cells and ganglion cells, and then off to the nerve, off to the optic nerve, then to the brain. And so basically for the photoreceptors, we have the rods and cones. In the, like basically in the middle, in the center, in the foveal center, we will have more cones because they are more, I would say, like focus on acuity and stuff like that. And then we have a bit outside of that, we start getting more rods, and there's definitely more rods than cones. And then what we get through that is we break it down into the visual pigments, which are the things that like help us to see these things. They're usually made in the inner segment, and then they're stored in the gels within the outer segment. And the outer segment is the part that like receives the photons, and that's what like goes from the light, and then we go through it to the nerves and like synapses and stuff like that. So the visual pigment is made out of 3 types. There's the opsin, chromophore, and then there's the melanopsin. So the opsin is a long protein, and it's usually like just one protein. And then there's chromophore, which is like just sticks out of it, and it's usually like this when it's not activated. So it'll be in the 11 cis position. And then once it photo activates, it isomerizes, which means it changes shape, and it goes like this and becomes all trans. However, that's just for the chromophore and opsin, and they're together, and they're found in either rods or cones. And melanopsin is a different part. Melanopsin is more for circadian rhythms. And so back to opsin and chromophore, they're mainly focused on light, and for rods it would be rhodopsin, and then for cones it'd be photoopsin. And that's how we get transduction, which is that whole process of light hits the external segment, which is where the visual pigments are stored. And then we get the like photoactivation, which goes from cis to trans. And then we get the hyperpolarization, which means that there's like the voltage is basically starting. And then once we get to that, this is where we view the graded potential, which is basically like if those are activated up top, there's going to be less neurotransmitters or glutamate produced, and like just signal to be produced. And here's a chart, I'll post it on the thing.